Hello everyone, welcome to our final Unit 2 video lecture, which is going to be on the macromolecule of proteins. Alright, so let's get started here. Alright, proteins are going to be one of the largest polymers that we've talked about this whole entire unit, even so sometimes bigger than what you would find with cellulose, starch, or glycogen. Now these polymers are going to be made up of carbon, hydrogen, oxygen, nitrogen, and the S stands for sulfur. So we've had your CHO with a 1 to 2 to 1 ratio. We've had your CHO with no ratio. We've had your CHON-P. And now, what do we got? We got your CHONs. Alright, kind of sounds like a Z at the end, but we have your CHONs going on right now. So don't forget it. Carbon, hydrogen, oxygen, nitrogen, and sulfur. And again, there is no ratio between what these elements would be in any particular protein. They are essential for life, so every single thing that is going on in your body is pretty much the direct result of a protein. Again, you can see where you get some of your proteins from up here: chicken, nuts, fish, cheeses or dairy, you name it. All right, all of these items will still have protein, but you got to be careful because depending on how much red meat and chicken that you're eating, for example, you might also be consuming excessive amounts of saturated fats and cholesterol. Now, the building blocks for proteins are going to be called amino acids. We've mentioned these before. So you're going to have amino acids that are made up of CHONs, and there are, at least for living things, 20 different types of amino acids that we can use to link together, as you see over here in this diagram. So take a bunch of amino acids, start to connect them together in a chain via a dehydration synthesis reaction. And eventually, once they fold together, shape dictates function, you will get some type of protein that will have a specific job. The bond that's going to link amino acids together, right, so right here you can see these little lines connecting to different colored balls. That is going to be a covalent bond, but the specific name that we're going to use is a peptide bond. So as soon as you have 3 amino acids linked together, you have what's called a polypeptide, just like we had a polysaccharide for carbohydrates. All right, so one more time, we have your CHONs. We're going to put those elements together to create the building blocks of proteins, which are amino acids. There are 20 different types of them. We don't have to memorize names, just know that there are 20. And then when we link them together via a dehydration synthesis reaction, the bonds that we create are called peptide bonds. Now, proteins have many, many different functions, and we're just going to focus on a couple of them. Here you can see what some of those are. We're going to create what's called enzymes to help with our digestive health, as well as pretty much run and speed up every chemical reaction in our body. They'll help to regulate the control and expression of our DNA and RNA. They help to move things around in your body, so they're used as transporters or messengers, just like red blood cells. They will have hemoglobin, HB, which is a protein on the surface which carries oxygen in your blood. Hormone production, support and protection of the body, so help to make ligaments as well as a component of actually making your bones. Antibodies for fighting against things like viruses and bacteria. And also, obviously, to produce muscle fibers which help for contraction and movement. So if we go through these again specifically, we're going to use proteins to build muscle, any type of muscle in your body, either skeletal, which actually will move your bones. You can also have cardiac muscle that moves, helps your heart pump, as well as some other varieties that we don't have to get into right now. Again, they'll help make up antibodies, which fight off infections. So right now up here, here's the dreaded coronavirus, right? If we have, I know some of you have had it, but if you have not been exposed, that means your body has not produced a specific type of antibody, which look like these little Y's here, that can recognize it, attach to it, and rupture its cell membrane and destroy it. So once we do get any type of coronavirus, it's not COVID-19, our bodies have to learn how to make these antibodies that will then recognize it and destroy it. And that would be for bacteria as well. Enzymes, which help speed up reactions in our body, just like this right here, will make sure that it'll take something which we call a substrate and make it undergo a reaction quickly and cheaper, not costing as much energy in the body. Messengers and transporters in the blood, like I mentioned, hemoglobin on red blood cells. production as well as regulation of DNA and RNA, major component of hormones, so estrogen, testosterone are made up of both proteins and cholesterol we mentioned in our lipids lecture. And finally, no matter how much hair you have on your body, hair is literally just protein. Your hair is not living. The only thing that's living is the follicle inside your pores that actually produces the hair strand. So all of these are just some of the functions that a protein could have. All right, moving on to amino acid structure. Each of the 20 amino acids, they're all going to have the same 3 key parts. Just one of them, though, may slightly vary from amino acid to amino acid, and I'll show you that momentarily. So the 3 key parts are going to be what we call an amino group. No matter if the amino group is on the left-hand side or right-hand side, it literally is a portion of an ammonia, which would normally be NH3. Right now, it is missing a hydrogen, so it's not called ammonia, it's an amino group. So you can see nitrogen with 2 hydrogens. This is the amino group. Every amino acid has it. Every amino acid also has what's called a carboxyl group. Again, either on the right or left-hand side, you're going to have a carbon double bonded to an oxygen as well as an OH group. This OH group here, as well as one of these hydrogens on the amino group, are going to be important for connecting amino acids. And then the third thing, and this is the one thing that every amino acid has but is going to vary from amino acid to amino acid, we have what's called the R group, or it might be called the side group, or even the variant group. This is different for every single amino acid. This could be just a hydrogen. It could be CH3. All right, it could be another NH2. All right, at that point, it really depends, but this is what can vary, no matter if you see it facing down or up. Every amino acid starts with a central carbon. They call it the alpha carbon. It'll have a hydrogen, but then the 3 key components that we're really interested in are the amino group, the carboxyl group, and what differs for every one of the 20 amino acids. We call it the R group, variable group, or a side group. Again, here's just another vantage point of what an amino acid looks like. I kind of like this one because it's showing you the electron pairs that we would have drew if we had been doing electron configurations, or using Lewis dot structures, I should say. Again, here is your amino group. Again, this nitrogen has a pair of electrons that are already formed. It's getting 1, 2, 3 electrons from these bonds. Same thing for this oxygen. It already has 2 pairs of electrons. All right, it needs 2 more to become stable, so it's getting those from over here. Again, basic structure here for the amino acids. Now, when we go to connect these, which once again will be done via dehydration synthesis reactions, what's going to happen is the carboxyl group of one amino acid is always going to bind to the amino group of another. That's always the way it works. You're not going to go amino to amino or carboxyl to carboxyl. It's going to be carboxyl group to amino group. And when you connect the 2 together, you're going to remove an OH from carboxyl, an H from the amino. You get water as a byproduct. And then you create the bond between the 2 amino acids, which we're going to call a peptide bond right down here. So connect 2 amino acids together, you get a dipeptide. 3 or more amino acids linked together would be a polypeptide. But once again, for every bond that you create, which we're calling peptide now, you will produce 1 water molecule as a waste product. And finally, again, going back to what we've been saying over and over and over again, shape dictates function. As you link amino acids together, they're going to take on one of 4 particular structures. Primary through quaternary. Primary structure is going to be characterized by a linear chain or sequence of amino acids, so you can see that up here. So once you connect an amino acid to another, you just kind of form a straight chain, no particular pattern or anything like that. However, once it gets long enough, it could take on a secondary structure, which is what we call either helical or pleated. All right, helical is a spiral. Notice those little dotted lines there. Those would be hydrogen bonds. That's what's actually making it take on this shape. Or pleated would be kind of little folds. Like if you take a piece of paper and try to make like a little paper fan, if you did that in elementary school, that's what pleated means. If the protein or amino acid sequence gets even big enough, it could actually fold on itself and produce a big glob, which you see over here. All right, that is a tertiary sequence. Again, it's one big strand of amino acids that's now kind of folded in on itself to be one big glob. And it might have some pleated as well as alpha helixes in there, but it's just one big blob now as a result. And finally, the largest of the protein structures, which you could even potentially see with your naked eye, are what we call quaternary. Quaternary would be when you have multiple globular sequences that all connect together. So in this diagram down here, you can see a red globular sequence, kind of like a purplish one here, as well as a white one over here. So you have three tertiary sequences all combined together to make one gigantic protein, and that's what we refer to as quaternary structure. All right, to finish out here for some review, again, multiple amino acids linked together in a straight chain, so this will be 2 because I see 2 R groups, would be primary. Secondary is going to be when hydrogen bonding between R groups starts to produce either a helical, spiral, or a pleated sequence. Tertiary is going to be, one more time over here, a big glob. And quaternary would be multiple globular or tertiary sequences combined together. So I have 1, 2, 3, 4 globs here together. And to finish out visually, again, if I look at these amino acids connected together here would be the R groups where my cursor is. All right, peptide bonds in between. All right, based on if the side change, the R groups are either polar or nonpolar, is what makes the chain of amino acids eventually start to take on either a linear sequence, like you see here, primary, or a spiral shape here, which will be secondary, start to spiral or get pleated back and forth. More interactions would cause it to be tertiary. Again, look at all those dotted lines. And then finally, multiple globular sequences coming together would be quaternary. If you have any questions, make sure you ask during our live lecture session. Thank you.